Buenas tardes a todos y todas.
Good afternoon, everyone.
Speakers, please turn the interpretation button to English. We're going to continue in Spanish. Bueno, es la segunda sesión del tercer día de la reunión.
This is the second half of the third day of our conference. I'm Deisha Kristen from Cancer Research and Biostatistics, the Statistical Center of SWOG, and founding member of the SWOG initiative in Latin America. I'm moderating this panel with Eva Bustamante. She's member of GOCHI, which is organizing the conference from Chile. I have some technical notes. Most know it, but for the newcomers, here they are. If you're calling from a phone without a Zoom app, you're just going to have audio, not video. We invite you to write down your questions on the Q&A icon. And if you can write them down during the panel members' presentations can help us organize during the roundtable. Please include the country from where you're sending the questions so that we can adapt the answers to your needs and interests. If you have connectivity problems, you can get closer to the server. You can connect the internet directly to your computer via cable, or you can log out and then log in again to the session. If there are English discussions and you want to change the interpretation language to English, to Spanish, choose interpretation and click on the corresponding language. We are recording the session. The purpose of the interpretation services is to promote a better understanding of the conference, but they do not replace the original. No person or entity will be liable for errors, omissions, ambiguities in the interpretation provided during the conference. All right, in the morning, we had Dr. Cuello speaking about financial challenges, financing studies in Latin America based on his experiences in Uruguay. Dr. Mueller explored the regulatory aspects in Latin America. And at the end, we had Dr. Höring. She gave examples of study design that use biomarkers in phase 3. In the second session, we will continue with translational and clinical research. Dr. Lopez will present some challenges as far as clinical research in South America. Then Drs. Grallo and Ray that will highlight opportunities to cooperate with SWOG and details on biobanks and opportunities to participate in translational research. At the end, Dr. Redman will explain designs for translational research that are after lung cancer using a study as an example. I'm going to give the screen to Eva now. Hello, good afternoon. Thanks, Deisha. For me, it is a pleasure to introduce Dr. Mercedes Lopez, our first panelist. Dr. Lopez is a surgeon, clinical immunologist from the University of Chile. She has a PhD in biomedical sciences. She has participated in several projects funded by different funds of early stages, which is so important. And this happens very little in our countries, particularly dendritic cell immune therapies for prostate cancer and melanoma. Besides, Dr. Lopez has supervised postgraduate students. So she has helped to create human capital. And she is also vice president of the Society of Immunology. Well, first of all, thank you, Eva, for your introduction. I think it's much more than what I deserve. It is very difficult for me to be able to present challenges, and I'm going to share my presentation. Well, the first slide will tell you what to do very briefly. It is just to show the reflections that come are born from the experience of basic and clinical teams to develop clinical protocols in Chile in phase 1 that include prototypes. Dr. Lopez, I'm sorry, we are not seeing your screen. There is a problem with the screen share. Well, I'll see what happens. Thank you. Can you see it now? Yes, now we can. And as I said, I will start with that summary showing that part of the reflections I will do is just like an attempt to understand or propose a hypothesis to explain why it is so difficult to do basic clinical research in Chile, from the first phases of research up to the first stages of phase 1 clinical trials. I want to dedicate this presentation to a person, a woman, a young PhD, Russian, Alexandra Elbakyan. She allowed us from her website to have free access to all of the publications. And I think it is quite fair to do so, since because of the experience of COVID, it is so important to have access to publications and data to generate knowledge together. Well, we have been working in Chile on this since 1998. We proposed an idea, which is active immune therapy for cancer. We work mostly with melanoma, using dendritic cells to immunize patients. And these dendritic cells, which are activated in a very special way using cell lysates. And with that, we can generate a vaccine prototype with which we started phase 1 studies in Chile that had very interesting outcomes. We were able to see that we could separate patients into two big groups, patients that responded to the active therapy we were giving to them. And there was a reduction increase in survival of responders and reduction in the survival of those who did not respond. The size of the prolongation of life of patients was quite interesting. So we thought that it was necessary to have phase 2 and phase 3 studies and see whether those outcomes were robust enough to be amplified or be validated with the studies with a larger number of patients. We published this series of first studies in different journals, and we have studied these dynamics of the dendritic cells with the lysate and the interaction with patients not only with melanoma, but prostate cancer and gallbladder cancer. And now we're going to start a phase 1 protocol using part of this development. However, we have been unable to go beyond. We have been unable to conduct phase 3 or phase 2/3 studies. And so we were not able to prove if those developments are appropriate or not. What I'm going to show to you right now is part of a reflection on the fundamental problems we face during this process. And the problems are two types of problems. The first one is the structural ones, systemic problems, which are shared by most of Latin American countries. And then, maybe this has already been mentioned in the sessions, that there are problems that are common, but you can manage them in a better way. First, knowledge about health should be tackled from knowledge in plural. Why? Because knowledge in plural are deposited, live in social norms and in objects. This means that scientific practices and the traditional logics of patenting cost this knowledge, and they started to be treated how the economy treats material goods. Clearly, this knowledge understood as this common practice of the society is not something material. You could explain this as with an apple, for example. I share my apple with you. You eat part of my apple. But if I share my knowledge with you, knowledge is not reduced. Most of the time, it increases. And this is particularly true in health, because health, understood in its complexity, has to do not only with the biomedical aspect, but also knowledge that includes public health, communication, education, and also traditional practices of the different communities. So if you say knowledge is not just one, especially health knowledge, there are several knowledge. It's knowledge in plural. And if you say that this knowledge does not have the same characteristic as the material things, knowledge is immaterial. So knowledge could be classified as something much larger. It is something common to all of us. When we think about it, to do clinical research in Latin America, we encounter big problems, big challenges, including structural issues and practices that we develop where we work and that we should change. One of the main problems is the unbalance between the different countries internationally. I will show this to you. I want to thank Gabriel Palma, Chilean economist, for lending me some of his figures. Please take a look at this panel. This is the productive matrix of Chile as compared to Malaysia. I will speak about Chile because I know it much more. The exports matrix of Chile is fundamentally raw material. But Chile doesn't only export raw material, but raw material with no processing. Our main exporting element is copper. It is exported just as a rock that includes all of the rest of the ores included in that rock. When we compare Chile with Malaysia, the productive matrix of that country is much more diverse. it is much more complex. And when we compare this with the work gap that is shown on the right, the productivity gap, Latin American countries that at the beginning had an increase of productivity. This is from Argentina, Mexico, and Brazil. Chile is exactly rather the same from the 80s. As their economies have simplified and the productive matrix too, this productivity has fallen as compared to other countries of the world, which have maintained the trend to having a productive matrix that is more complex. As a corollary of this complex matrix of productivity, the labor has also become more complex. When we speak about clinical research, it is the capability to have a path from basic research, from the question that appears by the side of the patient bed to a product that can improve the situation of the patient. And that path is pretty much related more than with investment in science and technology. It is more related to a matrix of development that is complex, that includes the complexity of a country. If a country has a productive matrix which is just to exploit, it won't have any interest in devising biomedical devices or biotechnological elements to generate knowledge in the country, so as to have this value chain. However, despite all that, Chile has levels of productivity that are very interesting. For example, here on the right side panel, I show you the number of papers. Those figures are in Spanish. And here we have the number of papers that we have in Chile in relation to the economically active population. These are Western Europe countries, Asian countries, Middle East, North America, and this is Latin America. These are our countries, and there is Chile. Chile has a percentage of publications that is interesting per million inhabitants that are economically active. So this represents a development of basic research that is interesting, and that gives us hope that we can have clinical research of high level in the future. Chile has something very interesting. Clinical teams have been able, despite all difficulties, have been able to do research, a series of academic projects which have dealt with a series of health problems in the population. But there is a consensus that we still have a long way ahead of us to achieve results that are beneficial for the population. So the problem of financing. We have a serious problem here. If we believe that knowledge is common because of its immaterial nature, so there is no possibility for it to be treated with the same rules or laws you use for material goods, we can see that what has happened in the biomedical research, but not only there, that financing of biomedical research has started to become privatized. Funding comes from large pharma industry, pharmaceutical companies that outsource clinical research through the CROs. This type of funding has different problems. Number one, and the fundamental problem, is that those pharmaceutical companies and the way to have private financing is focused on clinical trials that we try those drugs, but the basis doesn't stay there. So we cannot develop human resources or the necessary know-how for us to be able to do the full development in our countries. This is a fundamental problem because this has to do with the way in which we understand the response to the problem, the response to the different healthcare problems in our population. Well, there are other questions that are more important, which is what is the relationship that those pharmaceutical companies and CROs have with the different communities where clinical research is being conducted? What are the benefits for those communities? What are the rights communities have when clinical trials are developed? And the most fundamental thing for us in this event is how this stimulates creativity in research. How can we take part in those developments of the pharmaceutical companies? How can we share knowledge? How can companies share knowledge with us, share results and data that is being obtained in our region? How can we become active players, not mere recipients? The contradiction lies in how, from the standpoint of the academia, the academic clinical research, how can we take control? How can we get a hold of the clinical research done in the country so that we can get in touch with peers in other countries in equal conditions? Thank you. I'm going to finish very quickly. The other fundamental problem is this way to understand the system, this privatizing circle. The other problem is the molecular character of our research. We just generate biomedical knowledge without thinking of other things that are around the health problem. And this is more and more evident internationally because the size of the investment in health that doesn't go hand in hand with the health results in the population. We see an increasing gap between the investment in biomedical research and the impact that investment is having on the health of people. And finally, and very briefly, I want to show you this figure where you can see the high scientific productivity that we have despite our very small funding. Sometimes we don't know the real capabilities we have to do clinical research and for our population. Up until 1980 in Chile, a series of vaccines were produced. We didn't only vaccinate our population, but we exported those vaccines to other countries of Latin America. For example, the rabies vaccine to Brazil. Since 1980, and with the idea that those services should be bought outside since they were cheaper, the infrastructure to produce vaccines at the Bacteriological Institute, now Public Health Institute, started to be dismantled. But in the year 2002, it was fully dismantled. And today, we cannot develop vaccines in Chile. I didn't have enough time, but these are interesting data of 2012. These are the studies done in Chile. These are the studies of foreign labs. These are the foreign CROs, which account for 90% of the clinical trials done in Chile. National labs and others have much less. What can we do? Fora like this one are so important because through academic exchange, we can discuss with clinicians, academics from other countries in equal conditions to see how can we achieve synergy between the academia outside the country and the one that is done in our countries. But we cannot deny that we need a deep conversation. We need to discuss the matrix for development in each one of our countries to understand that our economies and the well-being of our population in health or any other aspect will be determined by making our economy more complex, understand that we have the necessary capabilities to develop biotechnology in the country and be able to share our knowledge with the rest of the world by including communities, territories, and communities with different cultures in Latin America. Thank you very much, and I apologize if I didn't keep the time. Thank you very much, Dr. Lopez. If you have questions for Dr. Lopez, please write them down on question and answer. At the end, we're going to discuss the questions. Well, the next presentation is by Dr. Julie Grallo and Dr. James Ray. Dr. Grallo is professor of medical oncology and global health at the School of Medicine at the University of Washington, United States. She's a director of medical oncology, breast cancer in the Alliance for Cancer Care in Seattle. She's the executive director of SWOG for breast and lung cancer. Dr. James Ray is executive director of translational medicine for SWOG. He is in charge of the operations of the biobank and biosamples of SWOG, and member of the SWOG Committee of Breast Cancer. He is professor of the departments of internal medicine and pharmacology at the University of Michigan, United States. The areas of experience and specialization of Dr. Ray include translational oncology with the identification and characterization of biomarkers, such as the expression of estrogen receptors and their implications in the prognosis and response to treatment in breast cancer. Welcome.
Thank you very much, and good evening to all of my Latin American colleagues. I'm sorry that we can't be face-to-face in the same room, but I'm hoping that 2021 we will finally get together again. Dr. Ray and I are going to overview some opportunities for clinical and translational collaboration from SWOG. And I'm going to talk about some opportunities within the US NIH's National Clinical Trials Network and SWOG trials. I'm going to briefly mention the SWOG Latin American initiative, the SWOG Clinical Trials Partnership, and then Hope Foundation opportunities. I know on Monday you heard from Dr. Blanke about the structure of SWOG, from Dr. Mohar about the SWOG Latin American Initiative, and from Joe Horn about the Hope Foundation, but hopefully I'll provide things in a slightly different way that will stimulate you to look out for collaborations between SWOG and all of you. I thought I'd show this slide that shows, at least as of right now, 2020, the different SWOG committees, because I think that's important, especially for those of you who are new to SWOG, some of the junior investigators, to understand that we have a bunch of primary committees, and these are the committees on the left that conduct research that run clinical trials. I'm the executive officer over breast cancer and lung cancer, but there are many other trials that conduct, committees that conduct trials. On the right are what we call our research support committees. And there's also the option of joining some of these committees. For example, there's a new one in immunotherapeutics. If that's something you're very interested in, you might see if you could join that committee and give input into some of the trials that they're talking about. There's a radiation oncology committee as well, and a patient advocate committee, and an adolescent and young adult committee. So take a look at all of these committees and see where your interests lie, and then think about attending the meetings at SWOG. Within the National Cancer Institute and the SWOG network, there are many roles that you could play on a clinical trial. You could be a study coordinator, also called a principal investigator. That would be the person leading the trial. There aren't a whole lot of trials anymore with limited funds and all to be the primary investigator, but there are lots of places where you could be a co-investigator on one of these studies. You could lead a sub-study, such as something involving quality of life or patient-reported outcomes or translational medicine studies. So there's room not just for one principal investigator, but for multiple questions being asked and run by co-investigators or sub-investigators. There's also the opportunity to look at SWOG trials that have been completed and evaluate the retrospective data, the specimens. Dr. Ray will talk about that, and ask scientific questions about trials that have already been completed. And then there's the opportunity, and we have some unique funding from the Hope Foundation for pilot projects where you just want to get a little more data so that you could potentially propose a new trial to SWOG. Now, we know about the SWOG Latin America Initiative. That's what's brought us all together. This is a program to develop cancer clinical research infrastructure across Latin America. The initial collaborations began as far back as 2004, and the Hope Foundation began supporting the SWOG Latin America Initiative in 2009. So last year, we celebrated what we called the 10-year anniversary in 2019 of the SWOG Latin America Initiative. And the key reason that this initiative was formed was to address cancer questions unique to Latin America. The etiology of cancer, biology, clinical presentation differences between Latin America and North America, looking at different drugs and assays and technology and health systems and cost, with a particular eye to Latin America and the cancers that occur in Latin America. You've probably seen information about this study. This is the first study within the SWOG Latin America Initiative. It was built in Spanish. It was intended to include participants from Mexico, Peru, and Colombia. It has aims to characterize the expression of immune checkpoints in gastric cancers in Latin America. And actually, CRAB worked to create a new registration application just for this particular project, as well as all future SWOG Latin America Initiative studies. The SWOG Clinical Trials Partnership is another very important piece of SWOG. It's separate from the money that comes from our U.S. National Cancer Institute. So this is not government funding. This is funding predominantly from industry that allows us to do a separate set of research studies. It goes back as far as 2008 when we had something called the SWOG Clinical Trials Initiative, and it was created as what we call a limited liability company under the Hope Foundation. And it allowed us within SWOG to have the opportunity to run non-federally funded studies in parallel to those that were supported by our US National Cancer Institute. In 2018, we expanded our strategic collaborations with industry through what we call a Preferred Partners Program and renamed it the SWOG Clinical Trials Partnerships. And for SWOG Latin America Initiative members, this offers an opportunity for research that's not funded by the US government, but could be very important within the SWOG Latin America Initiative. Dr. Kathy Albane, who's also speaking at this conference, leads this initiative. And I believe that with proper collaboration with industry or philanthropy, international trials can leverage SWOG's infrastructure, our great biostatistical group, our great translational medicine group, and maximize the information and the efficiency of such trials. Now, the Hope Foundation, which you heard about on Monday, has many opportunities, and we have funded investigators, junior faculty from Latin America with many of these awards. So take a look on the SWOG website about these awards if they're of interest to you. We have the SWOG Impact Award, and this is an award looking at translating scientific discoveries to patient care, and it's based on innovation. We have an award called the SWOG Early Exploration and Development Award, or the SEED Award, and this is to encourage preliminary research that we hope to translate to clinical trials. We've got an award for secondary data analysis, where you can use the Statistical Center to help look at some database analyses. And we also have an award called the SWOG Trials Support, which will support a trial that's already been approved that needs some supplemental funding. SWOG has some great career development awards for all of you junior up and coming investigators. We have a virtual grant writing workshop, a Coltman Fellowship Program for early career investigators, a Career Engagement Award program for more middle investigators, a Young Investigators Training Workshop, which is 3 days, usually in Seattle, where you're developing a protocol and working with our Biostatistical Center. And we have the new John Crowley Award, a 1-month residency in clinical trial statistics with our Seattle biostatistical group. So in conclusion, I just wanted to send you to this really nice overview from the USNCI on perspectives on strengthening cancer research and control in Latin America that was published in 2018. And within this publication, it says that one strategy that has promoted longer-term engagement between the US and Latin America is ensuring that partners work on meaningful activities that add value to what the partners could do separately or on their own. So value added. Partners who understand why their involvement adds value are going to be more motivated and can justify to their internal leadership. So, you know, your own center, you're going to be able to justify the investment in your time and resources in order to conduct these activities. So let's work together. I just will give one little plug as I turn this over to Dr. Ray for an upcoming 9th annual symposium on global cancer research sponsored by the USNCI Center for Global Health that will be March 10th through 11th, and abstracts are being selected now. This will of course be virtual, but I think this is a great conference and some of you may have great abstracts to submit. And I am going to stop screen sharing and turn this over to Dr. Ray.
All right, let's see here. So it's this. All right, can you guys see my screen? Can you hear me? And you can see
Yes.
it. Okay, great. All right, thanks. So yeah, I'll just touch base briefly here on the SWOG biobank and accessing the samples from the bank, as well as when you incorporate translational medicine components into your clinical trials. So actually this slide here, it's a bit dated. So the SWOG biorepository, we collect samples from a number of different, the disease groups, and the sample's ever expanding, and we're actually over 1 million samples at this time. And this is an incredible resource for individuals that are interested in doing translational research, because the quality of the clinical data that's associated with these samples, where you can test prospective biomarkers, both prognostic and predictive. So the goal of the biobank, and this is a separate grant that we have to store these samples, is that we're here as a resource for investigators to be able to, provide specimen collection and storage. And when you're writing a protocol and you're thinking about, the translational medicine studies you want to do and collecting samples for that, we can provide you with expert details on preclinical and pre-analytical protocols, so that the samples are collected properly and so that they can be used in future use. And so we implement the best practices in biospecimen sciences, both at the collection, processing, storage, and distribution. Some of the services that the biobank provides, it's a full service bank where you can provide pathological review of slides. We're doing a lot of work now with slide imaging for the real-time pathology. And this is actually ramped up quite a bit due to the global pandemic. The bank can also provide experts in the central processing of samples, which includes making tissue microarrays for biomarker studies, nucleotide and protein extractions, molecular testing, and a number of other state-of-the-art sample preparation processes. Now, the grant that we have to run the bank is a U-24, and this, in a nutshell, what this pays for is for collecting the samples and storing the samples. So it doesn't cover the cost for the actual translational medicine studies that you wish to use the samples for, and that you need to go find funding for. And as Julie had mentioned, there's a lot of opportunities through SWOG, especially with the Hope Foundation, for doing these studies. The bank is in charge of collecting samples from the phase 3 studies, as well as the randomized phase 2, and the samples are stored at our biorepository, which is at Children's Nationwide down in Columbus, Ohio. So the treatment grant and this comes to do with how the money is distributed for sample collection and future use. The treatment grant pays for samples from the large phase 3 studies, and historically it was only the randomized phase 2 studies for sample collection. However, we have been getting new guidance from the National Cancer Institute, where phase 2 studies that are single arm may be eligible for sample collection. And that's especially important in rare diseases. And that could be very representative of some of the studies that would be utilized from our South American colleagues. And so we have gotten into the process now of every time we ask the NCI if we're able to use our grants to collect samples from rare diseases. And almost all the time now, we get a yes answer for that. So as I mentioned, I won't go through all of this. The bank and SWOG is in charge of collecting the samples, but the investigators are in charge of finding the funds to be able to actually run the assays, you know, the biomarker assays and the molecular profiling assays.
5 minutes remaining.
As Julie had mentioned, there's a lot of opportunity through the Hope Foundation, and I actually run the Impact Award mechanism for the Hope Foundation. And we also, as she mentioned, the seed grants. In addition to that, there's opportunities through the NCI, and this is the biomarker imaging studies, for studies that you can you write proposals and access funds to be able to do the translational medicine assays. So when one is thinking about doing a translational medicine proposal, there are two types. There's the prospective that gets written into the protocol that's under development, and then there's retrospective studies. The retrospective studies are the ones that are using samples that have already been collected and are stored in the bank. And I've been involved in a number of these studies, as I have a lab here, and I do my own translational work, both within SWOG and with a number of other cooperative groups. So in the case with SWOG, for accessing samples, and as Julie had mentioned, sometimes you want samples so you can generate pilot data to justify an additional clinical study or to actually, you know, test your biomarker of interest. And the process here is a very simple 3 to 5 page proposal, basically providing a justification of why you want the samples. And a key point here, and we've heard from Mary earlier, is that when you're writing these proposals, is that you know the science, you know the assay, you know the hypotheses you want to test, but you have to, I highly recommend you have to have the input from our wonderful statistical colleagues. That is what is crucial for justifying accessing these valuable clinical samples. The details of what you want, again, this is where we can help you out because I review all these proposals that come in, and our statistical colleagues can help you out as well, and you can write a good, coherent, scientifically justified plan for accessing these samples, and then we will distribute them to you from our laboratory. With these proposals, we understand that sometimes you need a letter so that you can go get funding. And we say, provisionally, we accept you getting access to these samples. But to actually get access to the samples, you have to show that you have the financial support to do it, as well as we have to make sure that all the human research protection guidelines have been, are in play here. And I enjoyed the session 1 talk about and I'm interested in learning more from our Chilean colleagues about the human research protection IRB issues in Chile, because I've been sitting on Michigan's IRB for 10 years. And so I'm very familiar with the US rules, and I'm interested to learn more from my colleagues in South America. So
1 minute remaining.
So the review of these proposals, the written proposal gets submitted. It will then, I review it, and as well as my statistical colleagues, and then we submit it to the executive committee, which we call our triage, and it's discussed, and then we vote on whether or not to approve access to these clinical samples, and in the prospective setting, whether to, this is a viable and important translational medicine that should be included in the protocol. And with that, I am happy to answer questions at the breakout session. Thank you. I will stop sharing my screen if I can. There you go. There we go.
Muchas gracias a la doctora Gralo y doctor Rae.
Thank you very much. Now, we will have the presentation by Dr. Mary Redman.
Okay, hi, good morning. Can you hear me? It's not good morning, it's good afternoon.
Sorry.
Can you hear me?
Perdón. Sí, doctora Redman, we listen to you very well. I need to give the introduction about your presentation.
Okay, thank you.
Quiero agradecer en realidad la presentación.
I would like to thank the previous presentations. We were very attentive and taking note of all of the information that you gave us. I want to introduce Dr. Mary Redman. She works at the Huntington Cancer Center in Seattle, United States. She's the statistical leader of the SWOG Committee on Lung, and she is in the lung map trial. She has participated in 50 or 60 clinical trials for lung and other solid tumors, sarcoma, lymphoma, etc. She has a long experience. Her main experience is the design and follow-up analysis of clinical trials in cancer, and her area of interest is biomarkers as potential predictive markers. Thank you very much, Doctor. Remember that questions, please, should be asked under the in the icon of Q&A. Thank you.
Okay, thank you very much. As the speakers that preceded me, I very much appreciate the invitation to speak at this conference, and of course wish that we were in person in Chile. Today, I will be talking with you about statistical issues for immuno-oncology trials. And my experience of designing studies for immuno-oncology is mostly through my involvement in the LungMap trial. And that's partly what you see on the screen here is the background is our logo for the LungMap trial. I appreciate that Dr. Horing already gave you an introduction to the study, and so I will assume some knowledge, but it isn't necessary for your understanding of what I'll be talking about today. So the question that I'll be answering or discussing today, I should say, is whether or not immunotherapy has changed our design considerations for studying the treatment of lung cancer and designing studies in general. And while Dr. Horing described design considerations to include biomarkers in clinical trials evaluating targeted therapies, I would say that perhaps the elements for design of a clinical trial haven't really changed. And so I'll just put out this basic model here. When we're designing a clinical trial and to evaluate a new investigational therapy, we first need to define the patient population that we're going to be looking at. And then once we have that defined, we think about the endpoint that we want to use to evaluate the therapy in that population. Of course, that'll be related to the stage of treatment, the phase of treatment, and what information we have preceding that. And once we have that information, we typically then define the effect measure or the approach that we're going to use to evaluate the endpoint in the population to determine if this investigational therapy has activity or should be pursued further. So my experience evaluating immunotherapies in lung cancer has been, as I said, within the LungMap trial. And as Anja described, the LungMap trial evaluates both biomarker therapies, and then for patients who don't have one of these biomarkers, they are allowed to enroll onto what we call our non-matched trial. And our experience to date within the LungMap trial has generally been evaluating immunotherapies in this so-called non-matched population. The first study that we conducted in this space was a study in second-line advanced non-small cell lung cancer patients with squamous histology, and we randomized initially the patients to receive durvalumab, which is an anti-PD-L1 therapy, against docetaxel. As was mentioned, the trial was initially opened in June of 2014, and it was only then in March of 2015 that durvalumab nivolumab was just approved as in the new standard of care in this setting, which is the same class of drug as durvalumab. So we had some data on combination of a randomized study, but we also transitioned the trial to be a single-arm trial. So I'm presenting here to you the results from that study. On the left-hand side, you can see the experience from all patients receiving durvalumab. And the median overall survival was 11.6 months with a confidence interval of 10.2 to 14.3 months. On the right-hand side, you can see the experience when the study was randomized, a subset of those patients. And the median was 12.1 months in the randomized population receiving durvalumab and 7.7 months for those receiving docetaxel, with a hazard ratio of 0.64 and a p-value for that comparison of 0.11. These data were quite consistent with the data presented on nivolumab in this same line and pembrolizumab in squamous patients. But, and so that was a beneficial and wonderful thing to see, but there's much more work to be done here. And one thing I wanted to note is that in our study, the response rate of 16% was not that impressive when you think about looking at targeted therapies. And we did some biomarker analyses which didn't show any type of association with PD-L1 expression or tumor mutation burden and benefit on these trials. This study has been previously presented and will be published soon in Clinical Lung Cancer.
Sorry to interrupt, Dr. Redmond. Could we change your microphone and try to put it on computer microphone to see if that fixes some of the sound interferences that we're getting? It's a little bit hard for the interpreters.
Yeah, I apologize. I partly wonder if it's my hair. Is it any better or is it still the same?
It's still the same. We can keep going. But
well, let me see if I can switch.
There. We're not getting any sound now.
Well,
oh, there it goes again.
I have to look at just a moment. There's it's harder when I'm actually in this presentation mode to see what to do. So let me just see. I don't know how to find my thing while I'm presenting.
Okay, don't worry. We'll keep going. Thank you.
Okay. Well, I apologize for my audio, and I'm also trying to figure out how to make this do what I want it to do. Okay, so hopefully the interpreters can hear me well enough to interpret, and I apologize for my quality of audio. The next study that we conducted in the lung map infrastructure was a study also in the second line, and actually second line and beyond, randomizing patients to receive nivolumab combined with ipilimumab against nivolumab, which was the new standard of care in this setting. The idea being that adding ipilimumab, which is anti-CTLA-4 drug, would enhance the activity of nivolumab and prolong survival in this patient population. The study was activated in December of 2015 and enrolled 275 patients. The study was stopped in April of 2018, and it was stopped due to lack of activity by the data monitoring committee. And you can see here the survival curves for this study. On the left-hand side is the overall survival data, where you can see that generally speaking, the curves overlap each other with some separation at the later points in time, a hazard ratio of 0.87, p-value of 0.34, and medians that were actually better numerically in the nivolumab alone arm. Progression-free survival was also not statistically different from each other, although more close to a significance level at 0.09 and a hazard ratio of 0.8. The response rates between the arms were almost identical, with a response rate of 17% in the combination arm and 18% in the monotherapy arm. And these results are significant, particularly as nivolumab and ipilimumab were recently approved of as first-line therapy. But yesterday there was data to suggest that the pembrolizumab and combined with ipilimumab may not be beneficial compared to pembrolizumab alone. So the story is continuing, and these data are incredibly important. So the question that I have is, are we studying the right population? There's some data to suggest that selection of patients receiving immunotherapy could be based on PD-L1 or TMB, but we didn't look at those, and most studies haven't necessarily selected on that. And so I'll further discuss that here. In the 1400I study, we have both tumor mutation burden and PD-L1 expression data. And the plot on the left-hand side here shows that there's basically no correlation between TMB scores and PD-L1 expression levels. The S1400I study is relevant and important in this area, in part because most studies have used PD-L1 as a selection biomarker and has done tumor mutational burden as a secondary biomarker analysis. And so our database is perhaps the largest database to evaluate this question. We first looked at whether or not there was a level above which there was patients benefited from the combination of nivolumab and ipilimumab relative to nivolumab alone. And on the left-hand side, you can see that the hazard ratio, which is the dark red dot, do not differ from 1 with their confidence intervals enclosing that one horizontal line. And on the right-hand side, you can see the association between PD-L1 expression levels and survival benefit between the arms. And there is no difference or no threshold above which the PD-L1 expression levels predict patients benefiting from the combination. We then evaluated whether or not there was a combination of TMB and PD-L1 levels that suggested patient benefit. And the highlighted in red, with we preset a selection of p-value of less than 0.1, suggested that for patients with PD-L1 expression of 0 or no detectable PD-L1 would benefit potentially with increasing TMB levels. And I'll describe that better on the next slide. On here, you can see the hazard ratios comparing across levels of TMB scores for patients with no PD-L1 expression. And once we get to above the 10.9 level for TMB, it appears that there's potentially a group that's benefiting from the combination of nivolumab and ipilimumab. And this is described better on these slides, where you can see the Kaplan-Meier curves, where the solid lines represent patients with TMB scores that were greater than 10, and the dashed lines represent TMB scores less than 10. The red lines represent patients receiving the combination of nivolumab and ipilimumab, and the blue lines represent patients receiving nivolumab alone. And if you compare the red solid line to the blue solid line, you can see that patients receiving the combination are doing better than those receiving nivolumab alone, with a hazard ratio of 0.37, representing a 63% reduction in the risk of death and a p-value of 0.02. Whereas the dashed lines, you can see that the blue line, being the nivolumab alone arm, is above the dashed line in red, with patients doing worse with the combination and a hazard ratio of 2.46. Whereas on the right-hand side, looking at PD-L1 expression levels of greater than 0, so those who are PD-L1 positive, there doesn't seem to be a difference between the treatment arms, but there's potentially a suggestion that TMB is a prognostic factor, in that the solid lines are grouped together and the dashed lines are grouped together. And so here's an example of discovery of a potential biomarker from an ongoing study when the biomarker question is not the primary question. We do have additional biomarker studies that are ongoing that are using these paired tissue and blood samples, which will evaluate multiplex immunofluorescence, nanostring, RNA sync, and O-link to evaluate other potential biomarkers of immunobiomarkers, I should say. So I talked about whether or not we're looking at the right population, and maybe it's that we need to design studies that can evaluate populations when we have a general question. So the next question is, are we using the right endpoint? If one looks at the series of immunology studies that were conducted in lung cancer from 2015 to, say, 2018, 2019, I think that we can learn a lot from those studies. On the left-hand side, those pink dots represent the difference in response rates between the investigational therapy arm and the control arm for a series of randomized studies, the CM denoting checkmate, the KN denoting keynote studies. So, and the OAK and Pacific being those studies evaluating atezolizumab. And so these are the key studies that have been reported in lung cancer over a series of years. And the point that I'd like to make is that when we were looking at targeted therapies, we saw huge differences in response rates that were clear that these targeted therapies were working. Whereas with the exception of the Keynote 189, most of the differences in response rates between these two therapies have been around the 10, at most 20% range, which is not compelling in terms of response being a measure of activity for these therapies. And on the right-hand side, you can see in green boxes that denote the hazard ratio for comparing progression-free survival between an investigational therapy containing immunotherapy and standard of care, and in the blue denoting the overall survival hazard ratio. Because progression-free survival is a shorter-term endpoint, we typically assume that we can that the effect size for progression-free survival will be larger, that we can make a bigger difference in progression-free survival. And we typically assume that we will see a smaller, I shouldn't say assume, we have observed typically these differences, that hazard ratios for overall survival are closer to 1. But with a few exceptions on this plot, generally speaking, the PFS hazard ratio is not larger than the OS hazard ratio, suggesting that progression-free survival might not be a good surrogate or intermediate endpoint to measure activity for these immunotherapies. So traditional endpoints such as response and progression-free survival are not good endpoints as an early read to assess activity of immunotherapy agents. The population, should we look at all comers? Should we do biomarker-defined studies? Should we look at our studies to be biomarker discovery? These are all questions that we need to think about when we're designing trials for immunotherapy. And then the final topic is do traditional measures of activity capture these treatment effects? And the key thing that we've seen in immunotherapy studies is the observation we have non-proportional hazards. And what do I mean by that? If you look at the plots on the right-hand side, you can see that in all 4 circumstances, the control arm of chemotherapy is doing better than the immunotherapy arm at the earlier part of the curve. So between 0 and 6 months, let's say, and that later on events, patients do better with the immunotherapy drugs. Now, the curves don't cross at the same point, but there is some suggestion that there's crossing. And so non-proportional hazards suggest that the relative benefit of the immunotherapy drugs isn't the same across that whole patient experience.
4 minutes remaining.
Thank you. Now, this is not a new phenomenon, and in fact, we saw this with targeted therapies. The panels shown here show the survival curves and progression-free survival curves for gefitinib, an EGFR targeted therapy, compared to platinum-based chemotherapy. And they show that if we did an unselected patient population, that the curves would cross, whereas if we looked only at patients who had the EGFR mutation, the curves separated quite nicely, which could suggest that perhaps that's what we're seeing with immunotherapy trials, that there's a biomarker that's a predictor of benefit. The only problem is that these biomarkers are less clear-cut as they were for targeted therapies, and we have more work to do. So a little bit more on proportional hazards or the lack thereof. The left-hand side curve shows what proportional hazards would look like. The relative benefit for the investigational therapy relative to the standard of care is the same across the whole time that we're observing patients. Whereas on the right-hand side, this is what we would call a time-delayed treatment effect, where the curves are the same up until 6 months and then separate. The key thing to note is that standard approaches that summarize survival data with hazard ratios and compare the curves with log rank tests are typically averaging over the entire experience of those curves. And if there's no difference for a period of time, there's a time where you're averaging over no difference. And so there's most likely to be low power to detect the difference. We could do things such as evaluating the differences in the curves at different point estimates, such as was denoted by the blue dashed line showing if we looked at the point estimates at 6 months, or in the green dashed line at 12 months. But that requires us to pre-specify when the curves are going to separate and what is a clinically meaningful difference. And so then there are others who have suggested that using a weighted approach, that maybe we are going to weight later events more strongly to look at the differences between the curves. Well, we did an analysis to evaluate the power of the comparisons. And what I will just summarize by these curves is that the take-home message is that a comparison of a percentage with an event at a landmark time is not very powerful. And it's better to use measures that summarize the full patient experience, such as a log rank test or a weighted log rank test. And we actually implemented this in a study that we have ongoing in LungMap called S1800, which is randomizing patients between pembrolizumab and ramucirumab as the investigational therapy to standard of care, docetaxel, ramucirumab, or other chemo monotherapies for previously treated and immune relapsed disease. And this study is actually about to complete accrual next week. We will be closing it to further accrual. And in this study, we used overall survival as the primary endpoint, even though it's a randomized phase 2, and we will be using a weighted log rank test that weights later events more strongly than earlier events.
1 minute remaining.
Thank you. So back to that question, I think that we need to think more fully about how we define our patient populations when we're looking at immunotherapy studies, and set ourselves up to be able to evaluate biomarkers to provide evidence moving into the future. We need to think about endpoints that are better at evaluating activity other than response and progression-free survival. And our design should consider the fact that immunotherapies might have different needs in terms of effect measures and how we evaluate the studies to be able to be powerful to detect differences when they exist. So to conclude, our study populations, I believe, should be designed with broader groups, and we should incorporate specimen collection and for biomarker evaluation and biomarker discovery. In terms of endpoints, we need better endpoints to assess activity, and perhaps things like ctDNA or imaging should be incorporated to evaluate for activity. And in the absence of a robust surrogate, perhaps overall survival is our best approach. And then in terms of evaluating effect measures and how we compare data, we should choose powerful and robust tests to plausible scenarios and use effect measures that are interpretable. Our greatest challenge in the research on immunotherapy is to design studies to be able to find features that describe patients that best benefit and what is needed to augment immunotherapies for those who are not going to benefit. So I thank you for your attention.
Muchas gracias, doctora Redman, por su presentación sobre diseños.
Thank you, Dr. Redman, for su presentation for designed for studies with biomarkers. Next, we're going to go to the round table.
Si tienen preguntas, por favor, agréguenlas bajo la pestaña preguntas y
under the icon question and answer.
Bueno, Eva, ¿quieres empezar con la primera pregunta?
Comenzar con la primera pregunta, que es para la doctora.
First question for Dr. Lopez.
Pregunta el doctor Retamales.
Dr. Retamales, how have you managed technologic development in a public university through national, international funds or public-private
I think that to answer that question, in the first place, we have to understand that financing to science and technology in Chile is extremely low. 0.38 of the GDP only is devoted to research. And we have different instruments to be able to finance research, but you have to compete for those. Public universities don't have their own funds to finance their own research, so we compete with the rest of the national researchers, both of the public as well as private universities, in order to obtain research funds. In general terms, private-public partnerships are scarce because the private companies in Chile do little research, even though in the last months regarding COVID, some initiatives have appeared. But in general terms, the private investment in research is very low. So what we do, traditionally speaking, is to compete for FONDEF projects, as we call them, for research and development, which allow us to finance the first part of the research, the development of prototypes, and sometimes arrive to a phase 1. But from then onwards, we have no financing.
Next question for Dr. Ray and Dr. Gralo. Could you please indicate where you can find a description of the data that exists for reanalysis?
Julie, do you want to field that or?
Well, I think we can both chime in. I think it can be found in so many places. So I guess my suggestion would be take a look at a trial. For example, when the publication comes out, and think about what additional questions could be asked out of all of that data that's been gained. So for example, I led the SWOG S0307 trial, which was a trial looking at 3 different bisphosphonates in early stage breast cancer to see if there was a difference between them in reducing recurrences or a difference in the side effects. So that publication is out, and I think we could all come up with other questions we'd like to know. So we could ask about the difference between pre and postmenopausal women. We could ask, was there, a difference based on whether they received chemotherapy or endocrine therapy or both? That's part of what we mean. There are clearly a bunch of samples. All of those samples that Dr. Ray mentioned, we could ask if we collected serum on patients when they entered and they had high levels of bone turnover, did that predict for who would recur in the bone? That's slightly different than just a pure data analysis. That would require a translational medicine piece. But there are lots of questions that could come up. And I think, they could be found almost anywhere.
Yeah, I agree completely with Julie. And, you start with the publication, the literature, and then to get more granular, you talk to the PI and the statisticians. And again, anything from when we do translational, it's a team effort, right? And so you need to talk to the PI, and this is why you come to the group meetings and you attend the discussions and you talk to the statisticians, and you can really flush out what samples are available to test your hypothesis.
Perfecto, muchas gracias. Una tercera pregunta.
Thank you very much.
Carla Vargas.
A question from Carla Vargas
López.
For Dr. Lopez. What do you think is the main cause of discrepancy between the biomedical development and its impact on the population? Should we change the focus of research?
Sorry. what I believe is that the first thing that one should do is to understand that our final target is to provide health to our patients or to individuals. In that sense, health is fundamentally a multiple knowledge or structure. They're separate knowledge. It's not only biomedical knowledge, it's not only the disease that conditions the health of the persons. And when you focus only on the biomedical knowledge, you lose part of the reality of the whole health problem. And that has a deep impact at the level of the population, because finally, we focus the molecule that might be useful to a certain population of patients, and we forget about all the other factors that have an impact upon health. The main example for us is the COVID-19 infection, which is the reason why we are online instead of being face-to-face. A pandemic can be solved in several ways. And one way of solving a pandemic is to have as an endpoint the treatment of the patients. We have to take care of the ventilators or the vaccine. But there's another way of solving this, and that has to do with prevention of the pandemic. How do we prevent persons from getting sick, from being infected? I think that we have lacked, and this has to do with the need for transdisciplinary approaches. We need other perspectives that allow us to understand that what we are doing with one molecule or one antibody or one biomarker fits in the context of a patient that has a social reality, that has other characteristics that need to be dealt with too. We should discuss this gap. The fact that the British are telling us that they are spending lots of funds in solving health problems, but that the impact that they see in their population is not being that important, that should lead us to rethink the way in which we are doing research in health. And I speak about research in health more than biomedical research, because I believe that biomedicine is part of a more complex system that has as a final horizon the health of persons and not only treating the disease. And that involves a question to Dr. Miller, related to a question to her, that we have to understand that health is not only the relationship that we have with other researchers from other non-biomedical areas, but also the relationship that we have with the communities and individuals. Health data are our main output, are the main input to, the main material that is telling us whether what we are doing globally is well targeted or not. As a system, I mean. As a system, we should look at the data of our population, and we should see which is our impact. One example, and I conclude. In Spain, a paper was published showing that the diseases that have the greatest impact upon the people in Spanish are not the diseases that are more published about, say. And that difference, that gap, is the gap that in Latin America has to be solved, because we do not have many funds to research. We have little funds to research. We have a limited infrastructure to do research from the lab to the patient. And so our obligation is to respond to the health problems of our population. That is something we should try to assess more carefully. Thank you, doctor.
The next question for Dr. Redman. Which were the results of the SWOG-819. Did the design work?
Well, so the study was, we did not find a difference either in the FISH positive population or overall. And we did do some additional biomarker analyses that suggested that maybe a small subgroup of patients with squamous histology benefited from cetuximab. But unfortunately, well, actually, fortunately, now with immunotherapy, the question about cetuximab in the first line is probably not that relevant. So, but the design, it was a great design. We just need better drugs for my design. That's a joke.
Perfecto. Muchas gracias, Dr. Redman.
Thank you very much, Dr. Redman. The next question is about biobanks. I think it's for Dr. Ray. In relation to biobanks, do you have samples of Hispanic population?
For that, I don't know for sure, but certainly there are Hispanic and Latino in the bank, but it's certainly not highly represented. And that's what we want to do more of from the research side, because especially even from my own research interest, I'm interested in pharmacogenetics and genomics, and there's inter-ethnic variability in patient response. And that's why I think it's a wonderful opportunity with the South American initiative. But we would definitely need more. There are some there, but not enough for me that I think to do meaningful research at this time, and we want to expand those efforts.
Una pregunta respecto a
a question about the request for funds. The request for research, the translational research funds. Do you need to get in touch with CRABS biostatisticians beforehand, or do you get in touch with them once you've gotten the funds?
No, you absolutely want to talk to your statisticians first. So when you write a translational medicine proposal to utilize the samples, to be able to get that proposal approved, you need the statisticians to justify it from a statistical power side. And so that's critical, and you need that. And then you can go get funding. So if you were to say you want to, as Julie had discussed, the many opportunities at the Hope Foundation, just before you submit that Hope grant, you're going to want to talk to the PI of the study and the statistician and have them help you write your translational medicine proposal. And once that gets approved, then you can go get funding.
Yo creo que la pregunta va más allá.
I think the question goes beyond statisticians. Should they be CRABS or local ones?
So my recommendation there is the CRABS, because they are the ones, Mary knows that study inside and out. You can talk to your local statisticians when you're trying to develop your hypotheses and after you've read the publications on it. But then to get granular and to really get down to putting together your proposal, you need the help from the SWOG statisticians. And Mary, maybe you can weigh in as well, or so can John. But no, you need the group statisticians to help you with it.
Yeah, and sometimes it's helpful we'll have a conversation where somebody has an idea and we can involve the study chair, the lung committee chair, and myself, and really discuss what we think is reasonable and possible, that we like to be collaborative. But yeah, we know what's possible. Sometimes we'll tell you that you have a great idea, but it's not possible given what data we have. And then sometimes we'll say, that's a great idea, let's go for it.
Exactly right.
How to do it.
Great. Y esa pregunta es de la doctora Müller para el doctor Rey, pero tal vez...
This is a question for Dr. Ray from Dr. Müller, but maybe other panelists can have experience with that. Is it ethical to include in informed consent a paragraph where the patient can authorize the use of his data or samples for future studies? If so, should we include that always?
Yes, so I recommend that. So there are two things that go into, when we're collecting samples for translational medicine. One, we have a hypothesis already set forth that we want to test. However, there are also additional hypotheses that we aren't aware of yet. And so we bank samples away for unspecified future use. And so you really, in the days in the past, we weren't as sophisticated in the informed consent in thinking about future aspects. And now we do, so that the patients have the opportunity to opt in or opt out for future use. And then, for a lot of molecular profiling that's going on in cancer, we need to make sure we have the proper genetic language in there and everything that we're following along with the common rule that we have in the US. And then I'm assuming in South America, and I want to learn more how much your common rule for IRB matches the US's common rule.
Okay.
Muchas gracias. Nos queda aquí una pregunta también.
We have one question for Dr. Ray. In Latin America, there is heterogeneity as far as the capability to store samples. There are centers that have just one refrigerator and other centers with biobanks formally constituted. If the sample is processed and stored according to the protocol of the study, is that enough to send the samples, or do you think that there might be initiatives to standardize sample management in centers?
So there's two things to that. Yes, absolutely. Part of the bank is we have standardized methods that just template language that goes into the protocol for how the samples should be collected and stored. As far as then about, whether you have the capacity to store large number of samples versus smaller numbers, it comes down to shipping cost. And that's how we have to work it out, because you don't want to just submit one sample at a time. It's better if you have the capacity to save a batch and send a whole box at once, because the shipping cost can become, you can start, especially with our international sites, the shipping cost can become quite high. And we want to, but we can help you with that as well when it comes to budgeting.
Thank you.
Bueno, creo que con
All
eso.
right, I think with this, we're going to close the round table. Tomorrow, we'll see you again for the first session. This is a different time in Chile at 4 p.m. Chile time. And tomorrow will be management of symptoms and palliative care. Thank you very much.